Hi, thanks so much for your interest in using Tractor. I hope this tutorial helps get you started in doing ancestry-aware GWAS on admixed cohorts. So here's just the brief outline. I'll start with some motivation, go into the overview of our statistical model, and then talk about implementation of the actual Tractor code. So to start off with our motivation, I want to reiterate a point that is now thankfully becoming common knowledge in the GWAS community. That is that the vast majority of our association studies are actually conducted on European cohorts. And if we look further at this breakdown of the small kind of wedge of the pie of who is not European, we can see that there's actually only a few percent from recently admixed populations, such as African American and Hispanic Latino individuals, groups that collectively make up more than a third of the US populace. And just to make sure we're all on the same page, an admixed individual refers to somebody whose ancestry is not homogeneous, but rather has components from several different ancestral groups. So there are actually many more admixed individuals out there whose samples have already been collected and genotyped or sequenced alongside phenotypes, but they're not making it into this figure due to being kind of intentionally excluded for being admixed. So there's really a pressing need for novel methods to allow for the easy incorporation of admixed people into association studies. So admixed people are generally removed due to the challenges of accurately accounting for their complex ancestry such that population substructure and stratification can seep in and bias your results. And in the context of GWAS, this means the potential for false positive hits that are due to ancestry rather than a real relationship to the phenotype. So in a global collection, such as this example from the PGC, even controlling for PCs, which is the standard way that admixture is attempted to be kind of controlled for, there's still so much variability in the data that there's a lot of concern over false positives. So because of this, researchers will often sort of draw a circle around people who are deemed homogeneous enough to include in the study and everyone else is excluded. So this nearly always ends up resulting in European individuals being included as they generally represent the bulk of samples that have been collected. So the main motivation of the project I'm talking about today was to try to rectify this issue and develop a framework to allow for the easy incorporation of admixed people into association studies. So with Tractor, we are handling this concern by directly incorporating local ancestry. Local ancestry tells us the ancestral origin of each particular haplotype tract in an admixed individual at each spot in their genome. So in this three-way admixed individual, the y-axis are the autosomes, the position along them is on the x-axis, and because humans are diploid, the top and bottom half of each chromosome is painted differently. And you can see that each tract in this individual is colored according to the ancestry from which it derived. So the intuition behind Tractor is that to correct for population structure, we effectively scoop out the tracts from each component ancestry to be analyzed alongside other ancestrally similar segments. So we do this by tracking the ancestry context of each of the alleles for each person at each spot in their genome. So note that here I'm only showing this red ancestry, but the same thing is going to be happening for the blue and green ancestry. The statistical model built into Tractor tests each SNP for an association with the phenotype by splitting up the ancestry context of the minor alleles. So in the two-way admixed example, we have our intercept, of course, and then we include terms for how many copies of the index ancestry there are for this person at that spot in the genome. So say if you have 0, 1, or 2, for example, African alleles at this location, as well as how many minor alleles fall on your ancestry A versus ancestry B backbone. So those are the first three X's in these terms. And this is what corrects for the fine scale population structure. If there were differing allele frequencies for the ancestries at this spot in the genome, as we expect there to kind of routinely be due to different demographic histories of modern day human populations, then we're no longer going to be confounded by this as we've sort of deconvolved them. So the minor alleles seen on each ancestry backbone are kind of properly scaled to their background expectations. Note that while I'm showing a two-way admixed example here, this model can also scale easily to an arbitrary number of ancestries by addition of terms, as well as allowing for the ready inclusion of all of your necessary covariates. So I already went over how our model corrects for the fine scale population structure at the genotype or haplotype level, which is what's allowing the inclusion of admixed people in a well-calibrated manner in GWAS, but it also has some other nice benefits I'd like to briefly mention. We've built in an optional step to recover long-range tracts that we find to be disrupted by statistical phasing by using our local ancestry information. And with respect to GWAS, our novel local ancestry aware model can improve your results in a variety of ways, most notably through boosting GWAS power, generating ancestry specific summary statistics, and helping to localize your GWAS signal to be closer to the causal SNPs. So we really hope that this framework should advance existing methodologies for studying admixed individuals and allow for better calibrated understanding of the genetics of complex disorders in these underrepresented populations. So I'll now shift to giving a brief walkthrough of the steps involved in implementing the Tractor pipeline. So there are 3 different steps, the first one being optional for efforts that require complete haplotypes. I'm going to go through each of them kind of individually, but the first 3 steps are implemented as Python scripts. And step 3, the GWAS model, is sort of the recommended implementation is in the cloud native language HALE. There's a lot more documentation and description of these steps on our GitHub page, so please check out the wiki there for more information, as well as a Jupyter notebook to kind of walk through the steps of the pipeline. So before running local ancestry aware GWAS, you need to call your local ancestry. We recommend the program RFMix for this, which is described in their paper, Maples et al. 2013, as well as their GitHub repo, which I'm linking to here. So the success of Tractor really relies on good local ancestry calls. So it's really important to ensure that your local ancestry inference performance is highly accurate before you try to run a Tractor GWAS. Just to make sure we're kind of all ready to go, I wanted to show an example command for launching an RFMix run on 1 chromosome using some publicly available data. So this is sort of the basic parameter settings that we've come up with as being kind of ideal for our Tractor pipeline. Again, more details on the wiki page. But what I'd like to spend more time on is the actual Tractor scripts. So this first step in our pipeline is what's going to detect and correct for switch errors from phasing by using our local ancestry calls. You can see our supplementary information and extended data figures 1 and 2 in our manuscript for some additional context around what we mean by this. So this step uses RFMix ancestry calls as input and is implemented with the script unkink msp files.py, which tracks the switch locations and corrects them. So the output will consist of 2 files, a text file that documents the switch locations for each individual. So that'll be sort of your input MSP file name suffixed with switches, and a corrected local ancestry file, again, the same input name, but this time suffixed with unkinked, as in unkinking a garden hose, or kind of straightening out those switch errors. Here's your kind of example usage. You should just need to point to the MSP file name without this msp.tsv suffix and call this script. So this is one of the default output files from RFMix. Next, we also need to correct switch errors in our phased genotype file, and we're expecting VCF format for this. So this is what's going to actually recover the fuller haplotypes and improve our long-range track distribution. This step is implemented with the script unkinkgenofile.py and expects as input the phased VCF file that went into RFMix, as well as that switches file that we generated in the previous step. So the switches file is basically used to determine the positions that need to be flipped in your VCF file. Tractor also expects all of your VCF files to be phased, and it's recommended to strip their info and format annotations prior to running just to ensure good parsing. And again, step 1 is optional and won't affect your GWAS results, but can be useful for efforts that require complete haplotypes. All right, so in step 2, we extract the tracts that relate to each component ancestry into their own VCF files and calculate the ancestry haplotype and alternate allele counts for each component ancestry in your admixed cohort. So this step is implemented with the script extract tracts.py and expects as input the stem name of your VCF. You can have it just be unzipped, or gzipped is also allowed, as well as your, again, your RFMix MSP files. So you can input those unkinked files from the previous step if you chose to correct switch errors, or you can go straight from your original data if you did not. This script now accepts an arbitrary number of ancestry calls, which is very exciting. So you can specify the number of component ancestries with the --num anx flag. The default is 2. You can change it to however multi-way your population was. If your VCF file is gzipped, also include the flag --zipped. If not, leave that flag off. So there are multiple files output from this step, including the counts for each individual at each position for these 3 different pieces of information I'm listing here. So firstly, you'll get the number of copies of each ancestry at that spot. So if you have 0, 1, or 2 copies of ancestry A, B, C, however many ancestries are in your data. Secondly, you'll get the number of alternate allele counts on each of those ancestral backgrounds. And thirdly, you'll get VCF files for each of those ancestries containing only genotype information for that ancestry's tracts. So these will be sort of VCF files that will contain a bunch of missing data, will kind of blank out the other ancestries. And it'll also contain some half calls for instances where there's sort of heterozygous ancestry at a spot. All right, so our ancestry dosage output can now be used in our local ancestry aware GWAS model. The recommended implementation of the tractor joint analysis model uses the free scalable software framework HAIL. HAIL can be run locally or on the cloud. I'm going to be showing a cloud implementation here. And it can be useful to build and test your pipeline in a Jupyter notebook when applying it to your own cohort data. So for this reason, we supply an example Jupyter notebook written in HAIL on our GitHub, which can be adapted for user data. The specific commands to load in and run linear regression on our ancestry deconvolve file will be as follows. So here is the specific sort of suite of commands in HAIL that will read in and format our tractor dosage files just to make it a little nicer running things downstream. This is a little bit more involved in terms of loading files in than a standard VCF or matrix table. This is because we need to run GWAS on entries rather than rows. So for each ancestry and dosage haplotype file, we'll be kind of annotating that information in rather than just sort of one piece of information per variant. And this will become a little bit clearer in the following slides. But that's why there's like, a couple extra steps in this reading in dosage files portion. So next we'll join our ancestry and haplotype dosage files together onto a HAIL matrix table. So we do this by basically annotating the haplotype counts and any of the other ancestry information we have onto our first ancestry, ancestry 0. And here we're creating a new combined HAIL matrix table called MT, which will have all of this information in it. Basically, we'll have kind of our original ancestry 0 dosage information, and then structs for each additional term that we're annotating on. So here I'm showing examples for a 2-way and 3-way admixed example. In the 2-way, we're annotating structs for the ancestry 1 dosages, as well as our hap counts for our ancestry 0. So our index ancestry haplotype counts. These are both, all 3 of these are default outputs from our earlier tractor steps. So if you want to have more multi-way admixed populations included, you just need to add additional terms for each new ancestry. So 1 new dosage term for each additional ancestry and n-1 terms for the haplotype counts. So now we can load in and annotate our matrix table with our phenotype and covariate information. And from here on, we're now utilizing standard HAIL commands. The only thing to be careful about with loading in your phenotypes is to make sure that you're keying by the same ID field that will match the names in your genotypes file. And again, note that I'm doing this in the kind of Google Cloud example. So I'm pointing to the location of this file in my Google bucket. And finally, we can run the linear regression. So for a single phenotype, for example, total cholesterol or TC, as is shown here, along with our relevant covariates. So here, in addition to our intercept of 1, we have our half counts term and our 2 terms for our 2-way admixed population. So our ancestry 0 and 1 dosages. And then we also are putting in all of our covariates that we want here. So in this case, I'm using sex, age, blood dilution level, which is kind of a phenotype specific covariate, and then an estimate of the global African ancestry fraction that we got prior to this from using the program admixture. So we recommend putting in an estimate of your global ancestry in addition to these sort of local ancestry specific terms, just to account for if there's sort of a relationship to the phenotype with your overall ancestry proportions. And we find that the direct measure of global ancestry is a bit more accurate than PCs. So this is sort of the recommended strategy. So doing that, we are basically annotating each of the variants in our matrix table with the results of the GWAS. The results are again saved as a struct, here named TC, our phenotype. And this will contain pieces of information for each of the summary statistics that we listed here, including the beta effect size, the standard error, and the p-value. Within each of these, there's going to be an array of indexable values that's in the order of your terms. So the index of 0 is going to be your intercept. In the example I listed, prior to this, 1 is going to be your haplotype dosage, 2 is your ancestry 0 dosage, 3 would be ancestry 1, and then covariates would be 4 until however many covariates that you have. So you can pull out summary statistics for every term in your model and allow for kind of easy comparison. They're all going to be annotated into kind of one big struct that's just indexable by what position they were in your model. To run multiple phenotypes in batch, you can make a list of phenotypes first and then cycle through them, annotating the matrix table with multiple structs that will each be named the names of your phenotypes. So again, all of the information can be stored just in that one matrix table. In our Jupyter notebook, we also provide some code to generate Manhattan and QQ plots. You can select the term you would like to plot by its index, as we talked about in the last slide. And you can use the Hail commands plot.manhattan and plot.qq to visualize your results. So here we're pulling in our TC struct from the p-value. We want the third term, or the index of 2. And here is what those plotting commands will produce. So this is with those, the settings, by launching those commands, you'll end up with figures that look exactly like this. Great. So with that, I hope that this was helpful. Thanks so much again for your interest in Tractor, and feel free to leave questions on our GitHub repo or email me with any thoughts. I also have many people to thank, of course, for this project, including my KO1 funding from NIMH, and I'll also direct you to our paper published several months ago in Nature Genetics, which has a much more thorough description of our method and some other considerations. Again, our code is freely available up on GitHub alongside a wiki tutorial to help you get started with Tractor there as well. So thanks very much, and I hope that you enjoy using Tractor.